Absolutely. Here are 10 Puerto Rico Medical Board-style clinical vignettes focused on molecular biology, DNA, complete with 4 answer choices and detailed explanations. Vignette 1. A 5-year-old boy is diagnosed with xeroderma pigmentosum. He presents with extreme sensitivity to sunlight and multiple skin lesions. This condition is caused by a defect in which DNA repair mechanism? A. Base excision repair. B. Nucleotide excision repair. C. Mismatch repair. D. Homologous recombination. Correct answer B. Nucleotide excision repair. Explanation. XP results from a defect in nucleotide excision repair, the mechanism responsible for removing thymine dimers caused by UV radiation. Vignette 2. A child is diagnosed with Lynch syndrome, a hereditary cancer syndrome. Which DNA repair pathway is most likely defective? A. Base excision repair. B. Non-homologous end joining. C. Mismatch repair. D. Nucleotide excision repair. Mismatch repair. Explanation. Lynch syndrome, HNPCC, is caused by a defect in mismatch repair MMR, which corrects errors during DNA replication. Vignette 3. A researcher inhibits topoisomerase 1 in a bacterial culture. What process is most likely disrupted? A. Translation. B. DNA supercoiling relief during replication. C. mRNA splicing. D. Ribosome assembly hand. Correct answer B. DNA supercoiling relief during replication. Explanation. Topoisomerases relieve torsional strain during DNA replication. Inhibiting topoisomerase 1 prevents the relaxation of supercoiled DNA. Which of the following enzymes synthesizes RNA primers during DNA replication? A. DNA polymerase 1. B. DNA ligase. C. Primase. D. Helicase. Explanation. Primase synthesizes short RNA primers to provide a 3' OH group for DNA polymerases to begin elongation. Vignette 5. A mutation in DNA ligase would most likely impair which aspect of DNA replication? A. Initiation of replication. B. Formation of Okazaki fragments. C. Sealing nicks between Okazaki fragments. D. Synthesis of the leading strand. C. Sealing nicks between Okazaki fragments. Explanation. DNA ligase seals phosphodiester bonds between DNA fragments on the lagging strand. In eukaryotic cells, telomerase is essential in preventing chromosome shortening. Which type of enzyme activity does telomerase possess? A. DNA-dependent DNA polymerase. B. DNA-dependent RNA polymerase. C. Reverse transcriptase. D. RNA-dependent RNA polymerase. C. Reverse transcriptase. Explanation. Telomerase is a reverse transcriptase that adds DNA repeats to chromosome ends using its own RNA template. Aning. A newborn has symptoms of Fanconi anemia. Genetic testing reveals chromosomal breakage. Which DNA repair pathway is likely impaired? A. Mismatch repair. B. Homologous recombination. C. Nucleotide excision repair. D. Base excision repair. Correct answer B. Homologous recombination. Explanation. Fanconi anemia involves a defect in DNA crosslink repair, which is part of homologous recombination, a high-fidelity repair system for double-strand breaks. Vignette 8. Which of the following DNA polymerases is responsible for replacing RNA primers with DNA in prokaryotes? A. DNA polymerase 1, B. DNA polymerase 2, C. DNA polymerase 3, DNA ligase. Correct answer, DNA polymerase 1. Explanation. DNA polymerase 1 has 5' to 3' exonuclease activity, allowing it to remove RNA primers and fill the gap with DNA. During transcription, the coding strand of DNA has the same sequence as A. mRNA except T instead of U, B. mRNA except U instead of T, C. Template strand. Correct answer, B. mRNA except U instead of T. Explanation. The coding strand and mRNA are complementary to the template strand and have the same sequence, except RNA has uracil instead of thymine. Vignette 10. Which base pairing is correct in DNA? A. Adenine uracil, B. Cytosine adenine, C. Guanine thymine, D. Cytosine guanine. Correct answer, D. Cytosine guanine. Explanation. In DNA, cytosine pairs with guanine via 3 hydrogen bonds, and adenine pairs with thymine via 2 hydrogen bonds. Would you like the next set to focus on RNA processing, translation? Here are 10 Puerto Rico Medical Board-style clinical vignettes focused on RNA structure, function, and processing with 4 multiple-choice options and detailed explanations. Vignette 1, RNA types. A researcher isolates an RNA molecule that functions as the template for protein synthesis in the cytoplasm. Which type of RNA is it? A. tRNA, B. rRNA, C. mRNA, D. snRNA, C. mRNA. Explanation: messenger RNA mRNA carries the genetic code from DNA to the ribosome for protein synthesis. Vignette 2. tRNA structure. Which of the following best describes the role of the 3' end of tRNA? Binds to mRNA. B. Attaches the anticodon. C. Attaches the amino acid. D. Binds the ribosome. Attaches the amino acid. Explanation: the 3' end of tRNA ends with the sequence CCA and is the site where amino acids are covalently attached. Vignette 3. rRNA function. A drug targets the 60S ribosomal subunit in eukaryotes. Which RNA type is primarily found here? A. tRNA, B. 28S rRNA, C. 16S rRNA, D. 5S rRNA. Explanation: the 28S rRNA is a component of the 60S large ribosomal subunit in eukaryotes, important for peptidyl transferase activity. Vignette 4. RNA polymerase activity. Which RNA polymerase is responsible for synthesizing tRNA in eukaryotic cells? A. RNA polymerase 1, B. RNA polymerase 3, C. RNA polymerase 4. Correct answer: C. RNA polymerase 3. Explanation: in eukaryotes, RNA polymerase 3 synthesizes tRNA, 5S RNA, and other small RNAs. Vignette 5. RNA processing. Which of the following modifications occurs to pre-mRNA in the nucleus before it becomes mature mRNA? A. Poly-A tail addition. B. Removal of poly-U tail. C. Removal of ribosomes. D. Replacement of thymine with uracil. Correct answer: A. Poly-A tail addition. Explanation: mRNA is processed by 5' capping, splicing out introns, and 3' polyadenylation to increase stability and allow nuclear export. Vignette 6. Spliceosome function. A patient has a mutation affecting small nuclear ribonucleoproteins, SNRNPs. Which process is most likely impaired? A. Transcription. B. Splicing of pre-mRNA. C. Ribosome assembly. D. mRNA translation. Correct answer: B. Splicing of pre-mRNA. Explanation: SNRNPs are part of the spliceosome, which removes introns from pre-mRNA to form mature mRNA. Vignette 7. Ribosomal RNA synthesis. Where in the nucleus is rRNA synthesized and assembled with proteins? A. Nuclear membrane. B. Nucleoplasm. C. Nucleolus, D. Nuclear pore. C. Nucleolus. Explanation: the nucleolus is the site of rRNA transcription, mostly by RNA pol-wine, and assembly of ribosomal subunits. Vignette 8. RNA virus mechanism. An RNA virus uses its own RNA-dependent RNA polymerase to replicate. What is the major challenge the host faces in responding to such a virus? A. The host lacks uracil. B. The host cannot detect RNA. C. The host does not have RNA-dependent RNA polymerase. D. The host lacks ribosomes. Correct answer: C. The host does not have RNA-dependent RNA polymerase. Explanation: eukaryotic cells do not have RNA-dependent RNA polymerase, so RNA viruses must bring their own. Vignette 9. tRNA charging. Which enzyme is responsible for attaching the correct amino acid to its corresponding tRNA? A. RNA polymerase 3. B. Peptidyl transferase. C. Aminoacyl tRNA synthetase. D. Ribosome. Correct answer: C. Aminoacyl tRNA synthetase. Explanation: this enzyme charges tRNA by attaching the appropriate amino acid to its 3' end using ATP. Vignette 10. RNA stability. The 5' cap of eukaryotic mRNA serves which of the following functions? A. Promotes mRNA degradation. B. Allows tRNA binding. C. Initiates intron splicing. D. Protects mRNA from exonucleases. Correct answer: D. Protects mRNA from exonucleases. Explanation: the 5' cap stabilizes mRNA and facilitates ribosome binding for translation. Would you like the next